Hi, everyone. Welcome to today's webinar. My name is Bill Dempster, CEO with 360 Public Affairs, and working very closely with our friends at CanCertainty. And we've got one of the co-founders of CanCertainty on this panel as well, and I'll pass it over to him very shortly. I'd like to thank the supporters for this project, especially AbbVie, Amgen, Merck, and Pfizer. And I'll just get the slides going while we're at it. Start at the beginning. Okay, so today's CanCertainty webinar is on surrogate endpoints. We're going to try to work through the question, are drug review processes flexible enough to expedite patient access to new cancer medicines? And just Bob, over to you to just set the context a bit in terms of this Hot Topics webinar series, and then I'll help bring the agenda forward and the panelists.
Sure, thanks, Bill, and thanks for everyone here today for taking the time to do a bit of a dive into how our HTA and reimbursement systems need to evolve and adapt to the growing list of surrogate endpoints often used in trials for early stage cancer treatments. This is the fourth Hot Topics webinar. So far, we've covered the issue of PCPA and provincial listings with our webinar in November, where we reviewed the issue of time to patient and why timelines for public reimbursement of cancer medications are getting longer and how that impacts patients. In December, we tackled the issue of funding algorithms. And last month, we covered the issue of qualities or quality adjusted life years and how they're used in reimbursement decision-making and how arbitrary decisions on cost effectiveness thresholds impact patients' access to novel cancer treatments. So CanCertainty has developed this Hot Topic series so that patient advocates and other stakeholders are better equipped to participate in HTA and treatment access advocacy, and so that we can all work together to ensure that our public payers and HTA producers keep up with the rapid pace of scientific and technological change. So I hope you find these sessions helpful, and please stay tuned because CanCertainty is planning some more educational webinars for 2023. Back to you, Bill.
Thanks so much, Bob. And if people are not checking, that was on regular speed time. If you watch or listen to podcasts, sometimes I put it at 1.5. Bob can get a lot of information moving very quickly. So we might have to adjust sometimes. Sorry, I always rib you, Bob. My apologies.
No, it's true. I listen to podcasts at 1.25 and I tend to speak that way.
All right. So send in your questions, just a bit of housekeeping. We will do our best to get to them, questions and comments during the last 15 minutes of the webinar, and you're going to do that by using the chat panel button at any time to put them up there. And if you want one of the panelists in particular to tackle that question, please let us know who that is. We will be sending an email after this, and if we're missing any of the questions or if we needed to get to it, we'll send that note to all registrants so we can get to that offline in case we don't get to it now. So just in terms of the topic of today, we're going to learn a lot more about how non-traditional measurements for success in cancer trials are often called surrogate endpoints or non-traditional endpoints instead of the conventional overall survival surrogate outcome. And there are a couple of other more traditional endpoints. And these can include impacts on tumor, quality of life outcomes, and other treatment goals. We're going to learn a lot more about the different types of endpoints in clinical outcomes with our experts today. So many of these new or different outcomes are accepted by regulators, including Health Canada and the US FDA, to approve new medicines for use. However, these surrogate outcomes are sometimes not clear evidence of long-term benefits. And so provincial payers and health systems may experience challenges in adopting these new endpoints for their review and evaluation and funding decisions. So you're in for a treat. This panel of speakers has been working on and engaging with policymakers on this topic for many months now. And they've been refining their analyses and approach for much of that time. And I'd like to give a special shout out to Lisa Machado, who helped bring the patient perspective to the in-person conference at the Canadian Association for Population Therapeutics a few months ago, and helped set up a workshop at that conference, along with other experts on this panel. And our experts here and Lisa are planning to take these discussions to the international front. In May, they will be presenting on this issue at ISPOR in Boston, whereas we talked about earlier, it's Boston. And we're waiting on an opportunity to hopefully do the same at the CATA Symposium also in May, depending on how that agenda comes together in a couple of months. So to kick off the topic, we're going to have Dr. Christian Thorland explain what a non-traditional or surrogate outcome is and how it's generally used or applied, or in some cases not accepted as criteria to demonstrate clinical efficacy, or perhaps more appropriately put, patient-relevant efficacy. And I think something that we're going to pull out of this webinar is the terminology matters in terms of what we're looking for, clinically relevant, patient-relevant, et cetera. Christian will be followed by Tara Burgoyne of IQVIA and Lorraine Hudson of AstraZeneca Canada, and they're hot off the press research and analysis regarding two key questions. First, what does the oncology pipeline feature in terms of early stage cancer therapies and surrogate endpoints? And second, how has Canadian health technology evaluations, how have they handled surrogate endpoints to date for these cancers? And I know there's some early data, but it's so interesting. That report now is online. It's on IQVIA Canada's website. And that's another thing that we will get to everyone in our follow-up email to all registrants to this webinar as a link to that. We'll then kick off the discussion with Bob to help share his perspectives on what's important to the patient community regarding oncology endpoints. I'm also expecting the chat panel to light up with a lot of our experts on the line as well, because that's what always happens on these hot topics webinars. So don't be shy, get those questions and comments in as we're talking. So with that, I'm going to pass it over to Christian to get us started. Go for it.
Thank you very much, Bill. Okay, so I'd say with surrogate outcomes, it's a complex topic, and that's why we're talking about it. There's a lot of difficulties in making appropriate decisions around funding or approving new drugs, cancer specifically, but also other diseases. It's a lot of work to understand. I think we have quite the task ahead of us in terms of bridging the gap between various stakeholders, educating folks. That being said, if you look at what's at stake, it's definitely worth it. If you look at the literature, there's pretty good evidence that it's only about 20% of surrogate outcomes that actually show long-term benefit. And so if you're going to put all these resources into doing clinical research, clinical trials, post-surveillance studies, and you're not really measuring the right thing, then 80% of the time, then what's the point? And so we'd like to, I think, take a little bit of a step back here to begin with, and just go through the very basics, the kind of common sense cause and effect, if you will, what actually constitutes a surrogate outcome. What do we need to make sure is what criteria are met to be able to say that this is a good surrogate outcome, and similarly, which what does not constitute a proper surrogate outcome. So if you can go to the next slide. So in general, in cancer clinical trials, and this is maybe a bit more traditional, but just anchoring this in what's been done historically, a clinical outcome like overall survival is considered the gold standard. In many cases, we also have outcomes like disease-free survival, so the time that patient is disease-free, or at least progression-free survival. Again, if you look at the literature, these outcomes take typically a long time to measure. It's particularly the case for early stage cancers, where you have a lot of other issues, but overall survival is unfortunately not something that pops up right away. For that reason, there are estimates, I know, that it would take something like 17 years to run a clinical trial in early stage cancers. And that's simply something that we can't wait for. So we're forced to look at alternative outcomes that might be good correlates of these true clinical outcomes that we're interested in, whether it's overall survival, long-term disease-free survival, or just really good predictors of it. And so down on the lower left side is just a quick graphical illustration of if we have a surrogate endpoint, whichever way it's measured, we would like to see graphically and mathematically as well, some good correlation or ability of the surrogate outcome to predict the true clinical outcome. In this case, we put in survival as an example, but again, it could be any other long-term, very important clinical outcome. Next slide, please. And excuse me. So the definition of surrogate outcome is that it's a correlate of what we call the true clinical outcome, and that's necessary, but it's not sufficient. What it also needs to do is to capture the full net effect on the treatment. So we can go to, just skip one slide, it's just a picture. So one more, thank you. So when we think about surrogate outcomes, we think about a certain trajectory in the disease and criteria that have to be met. So the disease at one point might end up causing some surrogate outcome, which then later on in the trajectory causes the true clinical outcome. So let's say the surrogate outcome is a pathological response or radiological response, shrinkage of a tumor, and later on that predicts overall survival. Of course, for a surrogate outcome to be a good surrogate outcome, predicting or being correlated with the strongly correlated with the true clinical outcome, and for it to have meaning to use when we're looking at showing that a new experimental intervention is better than the standard of care, what we need to know is that the intervention impacts what happens between the disease and the surrogate outcome, and then that the surrogate outcome, of course, predicts the true clinical outcome. Next slide, please. The reality, however, is that it gets a bit more complex. And so I'd say for everyone here, just put your cause and effect type of thinking hat on. There are different causal pathways in the clinical trajectory. It's an infinite amount of different causal pathways in the trajectory of any patients, regardless of disease. And I think particularly with surrogate outcomes, we can break it down into a few standardized scenarios. So the first scenario here would show you scenario A, that the disease causes some surrogate outcome, and it also causes some true clinical outcome, but it's actually, there's no relation between the surrogate outcome and the true clinical outcome. And in that case, we wouldn't say that the surrogate outcome is a good surrogate predictor of the true clinical outcome, and we shouldn't use it in a clinical trial. There's pathway B, excuse me, where the intervention affects the pathway of the surrogate outcome, but it's not the only pathway. There's another pathway from the disease to the true clinical outcome. In that case, the surrogate outcome would show some sort of correlation with the true clinical outcome, but it's uncertain how much, and it may only be a moderately good or not very good surrogate outcome. There's, of course, pathway C, where the intervention impacts the pathway from the disease to the true clinical outcome, but not the surrogate outcome, but the surrogate outcome happens to also be a pathway that impacts the true clinical outcome. And so you can see with that cause and effect pathway, you might show mathematically that there's some correlation, but in fact, the surrogate outcome does not have anything to do, it cannot be impacted in any way by the intervention, which is really what we're looking for. And of course, we can have more complex situations where the intervention impacts both the true clinical outcome, the surrogate outcome, and other pathways. Next slide, please. It can get a bit more complex where, for example, we have the surrogate outcome that is a predictor of, let's say, survival or the true clinical outcome, but there's some confounding going on because of the presence of prognostic factors that predict both the surrogate and the survival. So they seem more correlated than they actually are. Next slide, please. And there's an even more quite clinically relevant scenario where the actual treatment of patients are impacted by the surrogate outcome. So a very common thing is that if you see shrinkage of a tumor radiologically, you may want to take patients off of treatment because you're thinking that it's clinical improvement. And that directly impacts the treatment pathway and therefore impacts survival in some cases perhaps negatively because you're taking off treatments that have worked. And so you can see it, things become quite complex. And these are all things that we need to think about when clinical trials are conducted and when they're being assessed for should new treatments be approved by regulatory agencies and should it be reimbursed by provinces, states, or other payers or stakeholders. Next slide, please. The last part, of course, is that does the surrogate outcome confer a improvement on the treatment over what is out control? So you may have that the surrogate outcome, that the intervention impacts the surrogate outcome, and the surrogate outcome impacts something like overall survival, whatever the true clinical outcome is. But when we're looking to approve new drugs, we're looking at how much better are they than some standard of care. And so if we see, for example, a 50% improvement on a surrogate outcome, does that also mean that from a new experimental treatment over control, does that also mean that there's a 50% improvement on true clinical outcome like overall survival? Are we okay with 30% improvement? Is that a sufficient correlation? And are those linked? So they're all things to consider when we consider surrogate outcomes. Next slide, please. Now, here's one example of HER2 positive operable breast cancer patients receiving adjuvant therapy with and without trastuzumab. And this is an example of where there's the slide I just showed you, where there's a relation between the comparative effect of the surrogate outcome and the comparative effect on the surrogate outcome and the comparative effect on the final outcome. In this case, you're seeing the lower two lines are disease-free survival, and you can see that the trastuzumab group or the experimental group experiences more benefit over time, and the dotted lines or dashed lines on top is overall survival, where you can see a similar trend, and you can see that trends occurs at the same rate approximately over time. So here's an example of where disease-free survival is a good surrogate outcome of overall survival. But no surrogate outcome, of course, is perfect, and so we need to make sure that we set proper bounds around that. What is the minimum requirement for what a good surrogate outcome, and at the same time not expecting perfection. Okay, next slide. Now, getting back to the lay of the land, and I haven't gone too heavy on this because there's a lot of interesting slides from my colleagues, so try to keep this a bit short, but there's a fair amount of interesting literature on unfortunately the fallacies of surrogate outcomes and how they're not used properly, and how they're drug improvements or drug reimbursements that don't happen when they perhaps should. There's some literature that clearly demonstrates that less than 20% of drugs approved on surrogate outcomes show long-term benefits, and that gets back to the first issue of why we really need to understand what surrogate outcomes are, how they fit into the causal pathway. Is it realistic to expect that some surrogate outcome is a good predictor or good correlate of some true clinical outcome or not? And should we actually be approving drugs? Should we be reimbursing drugs based on these surrogate outcomes? There's an unfortunate truth that most post-approval studies that are required after a drug is approved on surrogate outcomes, they just tend to use the same unvalidated surrogate outcome, and they don't even follow up for that longer. And in addition to that, when a drug is approved based on a surrogate outcome, there's a substantial drop in the clinical trial activity. So one example is out of 52 bevacizumab breast cancer trials that were ongoing, recruiting or planned, 10%, sorry, 10 of those, which is 21% of those, were terminated after approval, and 29% of those completed the trial, but the results remain unknown. So that's about a loss of 50% of data from clinical trials just because it was approved. And I don't know the reasons for why these were never put out there and why these were stopped, but it certainly is it's a trend that you can see across multiple approvals based on surrogate outcomes, and it's something that clearly it needs to stop, and there needs to be some more requirements around how much post-approval you should be enforced to do to, well, maybe not enforced, but at least to some extent required to validate the surrogate outcome after it's approved. Okay, next slide. The other unfortunate part of this, and this is particularly because we're honing in on early stage cancers, is that if you go and look at the literature and reviews of surrogate outcomes and their validations, there's quite a lot of literature reviews that basically systematic literature reviews of multiple validation studies, looking at what's the validity of surrogate outcome validation in different cancers and at different stages. And just a quick look at two of them that included several reviews with several clinical trials in each of them. So it's a total of more than 200 clinical trials across lung cancer and breast cancer. Out of all the surrogate outcomes that were looked at, there are only 3 trials looked at early stage cancer, and none of them looked at quality of life, and all of them only look at simple metric like the correlation, statistical correlation, and they don't go beyond that. So it is quite sparse what is being done in terms of properly validating surrogate outcomes, properly following them up, properly making sure that there's enough evidence to make appropriate decisions based on these outcomes. But they are necessary. So this is some work that needs to be done. So next slide, please. So these are the NMI slides. The next ones that Tara and Lorraine will be presenting are fascinating, yet a little disturbing. So they'll give you a lay of the land or the, in other words, the oncoming avalanche, as we call it, of use of surrogate outcomes in early stage cancers, particularly unvalidated or untraditional surrogate outcomes. So Tara, take it away.
Great, thank you, Christian. So we're moving a little bit into some data heavy slides for the next few. And just to set the scene a little bit before I hand it over to Lorraine as well. When we're talking about oncology therapeutics, we know that they have a significant contribution to the medicines in all phases of clinical trials in the Canadian pipeline. In 2021, it represented 1/3 of all medications that were to come. With the advancement of cancer care and ensuring that the patient receives the right treatment at the right time, it's important to understand the profile of the endpoints used in the current trials and to prepare for that potential impact on the HTA decisions. So to assess this potential impact, we conducted a study in which oncology clinical trials for early stage cancer specifically were assessed. Lorraine's going to talk to that a little bit shortly after I go through a couple slides, but to set the stage for what's to come, it's important to look at where we are right now as well. As part of the study, we also did a retrospective analysis to look at the historic review of oncology submissions to CADA. It's acknowledged that the type of endpoint evaluated is not the sole contributing factor and may not have substantial standalone impact on the recommendation outcome. But this assessment did consider multiple variables, including the statistical significance of the submitted evidence, the trial phase and design, quality of life, and economic considerations. So the objective of this analysis and what we're going to look at in the next few slides was specifically to evaluate the correlation between the primary endpoint that was used and the final recommendation that was issued by CADA. This first slide is looking at all oncology submissions across all disease stages where a recommendation was issued within the past 5 years. So this excludes any resubmissions, non-manufacturer submissions, or those for gene therapies. In total, we looked at 115 recommendations where they were reviewed and summarized based on the evidence submitted and deliberated on. So the solid bars are representing the submissions made to CADA and the ultimate recommendation for those that were containing traditional endpoints, which for the purpose of the evaluation that we're doing are defined as overall survival, progression-free survival, and objective response rate. So the solid bars, as I mentioned, are the primary clinical endpoints. In the shaded bars, those were where the primary clinical endpoint evaluated was non-traditional or the surrogate. So in other words, non-OS, PFS, or ORR. In this analysis, the majority of the recommendations were issued for adjuvant or metastatic cancer treatment. And looking at that pool, of all recommendations issued, only 13% were submitted and evaluated based on a non-traditional or a surrogate endpoint. Next slide, please, Phil.
You got it, Tara. Just a quick note and question for
Sure.
The acronyms. Would you mind expanding on those a little bit? And actually maybe take 2 seconds to say, okay, traditional endpoints are OS, and an example of a non-traditional endpoint is, go ahead, if you don't mind just taking a half second.
Sure, absolutely. So I think the most conventional traditional endpoint that we're used to seeing is overall survival, OS. And in this study, we branched that out a little bit to look at progression-free survival, PFS, and ORR, objective response rate as well. So when they're classified as the traditional outcomes, those are the three that we're looking at in this study. When we're looking at non-traditional, there's surrogate outcomes, we're looking at stuff like disease-free survival, event-free survival, metastasis-free survival. Those are still very survival-based in terms of the surrogate. There are others that we're looking at as well, pathological complete response and other sort of measures, which Lorraine will explore a little bit in her subsequent slides as well.
Excellent. I read the report. It was helpful to have a little glossary to go back to the different acronyms. So thanks so much for doing it verbally.
Great. Thanks, Bill. So now when we look at it, looking at the analysis specifically for an early stage oncology. So before we were looking at a broader pool of all oncology, including the metastatic stage. When we're looking at it from an early stage cancer, we see a very different picture than what we saw in the overall analysis. While this N value for this assessment is much smaller, there is a very distinct difference in the submitted evidence and ultimately the recommendation that was issued by CADA. This is positive, and it does demonstrate a degree of acceptance towards the non-traditional endpoints. However, in almost all cases, the surrogates that are used are still those that are based in survival, such as the DFS and EFS that I just mentioned. And it's important to note that most of these recommendations that were issued with a primary endpoint, surrogate primary endpoint, also had an accompanying traditional endpoint with them as a secondary outcome or measure. So what you'll see shared momentarily by Lorraine is that the type and frequency of those non-traditional endpoints previously evaluated on is not nearly as varied as what's coming down the pipeline. And on the right-hand side of the slide under the surrogate, you see that a lot of those are still based in the survival or survival-based outcomes. And we do see pathological complete response as well. So what exactly is that avalanche that Christian talked about? And what are the challenges that the industry and HTA are expecting to face in the coming years? So for that, I'll hand over to Lorraine, who will be discussing the clinical trial assessment and the potential implications in early stage oncology.
Thanks, Tara, and good afternoon, everyone. So as Tara mentioned, we had a bit of a snapshot on the experience to date, which I could, it seems pretty limited at this point in terms of how we really validate and how we, look at the evaluation process for surrogate and trials with surrogate endpoints. So we also wanted to take a look at in this analysis as to what does the future landscape look like? How big of an issue is this going to be? And so we did an analysis looking at all of the oncology trials that are out active between January 2017 and March 2022, where we started the analysis. We looked at trials that were in the phase 2 or phase 3 and that were active. And we limited it in this case to a solid tumor space with a single indication. You can see here that we really focused in on that early stage, non-metastatic, non-invasive stage 1 to 3 from a disease stage, because we recognize that the majority of those surrogate endpoint trials are really in that earlier stage setting. And so once we looked through those trials, we really took a deep dive into about 387 trials. And at a first glance, when we looked across the trials, again, this is early stage solid tumors, we could see when we look specifically at the primary endpoints, really about 70% of those trials had a single primary endpoint. And within those single primary endpoints, about 83% were surrogate outcomes. And interesting to note that within those surrogate outcomes as a single primary, about 40% of those trials did not include any traditional outcome as a co-primary or secondary endpoint, which is important because as we saw from the experience to date, generally there was that traditional endpoint as a secondary endpoint. So looking at this, I think right away we recognize that this is a very different picture than what we're used to seeing in the advanced or metastatic stage cancer trials. And so we know right away we've got something new that we're sort of contending with here. So if you move ahead a slide there, Bill. We also want to take a look at, across the 10 different solid tumor areas, how did this look up across each of the individual tumor types? And you can see here that while largely driven in the breast, lung, prostate, and bladder tumor sites, we can see from the blue columns or the light blue columns that really, surrogate endpoints are steadily increasing across all solid tumors. We haven't done the hematological space analysis yet, but I imagine it's starting to look very similar. So we know right away we have to start to figure out not only how we support the data, as Christian was mentioning, how do we support this data to ensure that patients at the end of the day will be able to benefit from these new treatments and the trials that are taking place. You can move ahead there, Bill. And interestingly, when we looked across, again, the early stage single primary endpoint trials, what was interesting is that what we can see is that there is an array of different surrogate endpoints, as Tara was mentioning. We're not really limited to a handful or 2 or 3 that we're trying to figure out how to support. It really is, dependent on what's appropriate for the specific disease type. And you can see here, if you look at, for example, pathological complete response, that's really the most common surrogate endpoint in current clinical trials at this time. And you can see even from a traditional endpoint like progression-free survival, that is more common, much more common than we're seeing the gold standard, which is overall survival in these trials. So certainly something that looks very different again as to what we're used to seeing in the advanced metastatic stage. So really, when we look at this, we know that evidence based on surrogate endpoints is increasingly being accepted to help decision-making at the regulatory level and in the clinical setting. But I think now we need to really start to pay attention on how do we explore to optimally support the use of clinically meaningful surrogate endpoints to increase the acceptance in access decision-making environment. You can move forward, Bill. And I think that's where, what brought us here today. I think this is the challenge that we all face. As Bob had mentioned, or Bill had mentioned, sorry, earlier, we are pulling together the data, the analysis that we've done over this past year, and pulling it together in a publication that is now posted on the IQVIA site. But really, this is intended to foster the conversation, which I think we're about to head into, but raise the awareness, foster the conversation, and see how we can manage together across stakeholders to help to solve for some of this. So with that, I think I can hand it back to Bill or Bob.
Yeah, thanks, Lorraine. And before we go on to Bob, your first question in terms of why is this important to patients? And Bob, you're welcome to tackle this too. We've unpacked the acronyms a little bit. It looks like pathological complete response is, the most common surrogate or non-traditional endpoint that you've shown in your last slide there, Lorraine. Can one of you give us an example of what exactly that means? What is pathological complete response, for example, in breast cancer or in lung? What would that mean to bring it down to patient terms? Who wants to jump on that one? I'm only asking because I think there's a lot of people on the line who probably don't have a science background, but are really interested in this because they're cancer survivors. And I don't know, Bob, if you want to jump in, you're welcome.
I'm wondering, I'm thinking we have an expert patient on the line, Deb Maskins, who I believe might be better equipped to describe what pathological complete response is. I call her an amateur oncologist from time to time. Deb, are you there? Can you give us an idea of what that is?
I have to elevate her to panelist. I'm promoting you, Deb. Heads up. You're coming in if you accept it. And good for you, Bob, for throwing one of our devoted attendees into a panelist mode if she's so inclined. While we're waiting for her, though, Bob, oh, here we go. Now I'm going to make you co-host, Deb, and you can do whatever you want. Turn your camera on and surprise guest panelist. Welcome, Deb. Thank you for joining us. Audio is really, really poor, Deb. I don't know if you're far away from your phone.
Okay, I'm getting closer.
No,
There
that's
we
good.
go.
Good. Okay, so I had some questions that I was going to type in, but can you repeat for me the question that Bob so very kindly passed my way?
Well, sure. I mean, basically, we're trying to look for a patient-friendly definition of what pathological complete response is. I mean, I know that basically it's the the absence of all signs of cancer in tissues under examination in the lab. But as somebody who's probably had a lot of tissues presented to the lab, I just wondered if you could give it a little bit of a deeper dive into that.
Well, okay, I think I can only partially answer your question, because I think what you're hinting towards, I am a patient and I have spoken to FDA Oncology about the use of these trial endpoints. And I think what we do know is that patients do not understand what these endpoints mean. In fact, patients, I would say, are deceived, because with so many of the names of these endpoints, you will see the word survival. So for things like disease-free survival, and we've done a bit of a survey of this with Canadian patients, patients should not be faulted for thinking that disease-free survival correlates with survival, when it doesn't always, and as it's been shown out quite often, doesn't. So I think coming to your question, Bob, the outlier on that is the PCR. And I don't think we know yet whether that is a patient relevant endpoint. You would think it would be, but I think there's a whole wealth of study that needs to happen when we come back to what Bill said, I think originally, about are these patient relevant endpoints? So are patients signing up for these treatments on a basic misunderstanding of what this endpoint means for them? And just to, I'll say this and then I'll stop speaking, but to one of Lorraine's slides, there was some discussion, and the one that's actually on my screen now, that talks about single primary endpoint. As a patient and advocate very involved in trials, I think we need to be thinking from a patient lens that patients expect more than a single endpoint. So for us to be designing trials in this day that are only measuring one primary endpoint, I would question, why is quality of life as low as it is on this graph? And why is quality of life not a co-primary in every trial that we do? Because it is equally important to patients. It's not just about survival. And I'll stop there and go on mute. Thank you for the opportunity.
Oh, that's tremendous. I mean, the quality of life, I think, is the one thing that, as a co-primary endpoint would really identify the patient value of these treatments. To Bill's question, why is this topic area so important to patients? I mean, from my perspective, I think what we have to be concerned about is that this is going to introduce, from the perspective of health technology assessment, a whole new level of uncertainty. And I think we have a lot of experience. And I mean, I know that we have some stats here that we've seen some drugs approved with some of these surrogate endpoints, and Deb alluded to one of them, which was an adjuvant treatment for renal cell carcinoma, which was approved using disease-free survival. But it was actually approved, and I believe it was approved because of some of the financial opportunity that was that this drug offered in the adjuvant setting. For the cost of 1 year of treatment for patients, there was a potential cure rate of a certain subset of those patients that really offered tremendous benefit to our health system from a financial perspective. Of course, that's the other issue, of course, is that there's going to be a whole group of patients that are treated in the adjuvant setting for this particular drug that I'm referencing that wouldn't actually need it. And when you're talking about an IO therapy, it's not without significant side effects and patient impact. So, but from my perspective, the issue here that we have to try to work through is to find mechanisms so that we can help resolve the uncertainty that these new surrogate outcomes introduce to our health technology assessment. And I just want to point out that I'm actually quite excited because last week, many of you might have seen in the weekly summary put out by CADIF that they've just announced a consultation on time-limited recommendations. So sometime in March, there's going to be they're working with the participating drug programs on a review on a process or a proposal for issuing recommendations that are time limited and contingent on evidence generation. Now, I think that this topic today is very important in the context of that upcoming consultation. So I just want to put it out there. But to me, this is the issue of the uncertainty it introduces. Of course, there's going to be a lot of drugs in this mix. They're going to be very, very important, some less so. But I mean, we have to work to resolve that uncertainty. And I think that's what this is all about.
Thanks, Bob. Maybe I'll bring it back briefly to Tara and Lorraine. One of the things that jumped out at me from the report was how CADTH is handling this so far. I mean, it looked like a relatively low number, but there are some early signs that CADTH may be struggling with some surrogate endpoints. Is there anything you want to talk about? I know the end number was like 11, and there were only 3 that actually came out where they said no to clinical because of X, Y, Z. And can you expand a little bit on that?
Yeah, I think, like I said, it is very positive. We've seen that these submissions that are put forward with the surrogate endpoints are receiving conditional recommendations are positive. Very few are receiving negative. Some of the ones that did receive negative is back to what we talked about. PCR specifically is one example that, there was a lack of clarity on whether or not improvements to PCR actually translated to clinically meaningful improvements in event-free or OS outcomes. So I think what is the key takeaway from this is that, yes, they're looking at it. They're looking at surrogates, but as I mentioned, right now they're sitting in that survival sort of framework, as opposed to what Deb was mentioning, the more patient relevant, or some of those that are even more disease specific. Some of the endpoints in these new clinical trials that are coming are very disease specific and not based in survival. So while it is very positive now, I think what we're looking for is what's coming down that pipe and how they're going to be able to make that correlation between some of these primary endpoints that they're seeing to what they're used to seeing in the OS or the PFS data that they're getting now.
Thanks, Tara. Lorraine, what's the industry doing about helping deal with this? And I don't know if you want to react to what Bob mentioned about how CADTH is coming up with time limited recommendations. Is that a signal back to the industry and the developers to make some tweaks in terms of phase 4s or other post approval studies or reviews?
Yeah, I mean, I think we all have a part to play, right, in all of this. And so I think part of it is trying to understand, what is the evidence needed to help create a stronger level of certainty. Maybe certainty is not the right word, but to reduce the uncertainty and bring some more comfort into managing through how we deal with these endpoints. We have to do a better job even defining the surrogate endpoints. We have to look at those validation instruments so that we can draw stronger correlations between surrogate endpoints and what we would consider more comfortable, maybe traditional endpoints. And so industry certainly has a role to play in this. I think, with the constant working with the real world evidence, we have to look at that and see how do we apply real world evidence. And then sort of how do we look at those time limited type approvals or agreements and how do we come back to that? So, but all of that, we can't work in vacuums. We can't work in isolation. We really do need to come around the table, all the stakeholders involved, including the very valued patient voice at the table to really impress upon not only the sponsors, but the regulators and the HTA bodies as to what is really important and what evidence do we need to make sure that this comes through and is accepted at the reimbursement level.
Bob, you had your finger up. Go for it.
I just want to say, so I mean, I recall when we were doing this whole workshop at the Canadian Association for Population Therapeutics at their annual conference last year, is that Christian had mentioned that there's going to be a strong need for HTA producers to develop and adapt their methods for engaging with these new endpoints, and that was kind of one of the big objectives that we had from CanCertainty, was to try to move our whole system forward. Right, it's not to like endorse, any particular endpoint or advocate for the absolute, acceptance of these endpoints as decent surrogates for survival. It's simply to, get us all moving forward to understand, engage, and develop and adapt methods for engaging with this. And I just wanted to say that we had a little bit of an interesting success in that regard. We were approached after that CAP session by somebody from CADIF, where they asked CanCertainty to host a special presentation on this topic exclusively for CADIF staff. I think we actually had some success. So on December 5th of last year, we did a very high, very deep presentation on all the issues that HTA producers and reimbursers have to start to wrestle with in order to to deal with this avalanche that's coming. So I think our a little bit of advocacy came out of this, I think, successfully moving this forward.
Maybe just
Thanks, Bob. Go ahead, Christian.
Just add to that quickly that I mean, in the other presentations we've done, there was a lot more statistics, and we sort of decided to take that out of this one. But the one we have at CADIF was very focused on methods, like what are they going to use for their decisions. And to Lorraine's point, also just having access to real world evidence, there's an adoption of that we not just look at the clinical trials, but we also look at patient registries, data vendors, larger databases, wherever you can get data from, because each patient data point really counts. And I think particularly with Deb's point about quality of life, there's a lot you can extract from that data to supplement what HTA bodies have to consider already. And so there's definitely a lot of moving pieces currently. It's early days, but it's I mean, I can almost assure it's going in the right direction. A little too slow, unfortunately, and that's why we have to keep pushing, but it's getting there.
So yeah, I've got two questions, and then again, thanks to somebody who threw something into the webinar chat. It looks very statistical, by the way, Christian. Maybe you want to jump on that in terms of an answer to the chat. So have a look there while I throw this out. What's the relationship between the surrogate endpoints and or I should say just the primary endpoints for many of these early stage cancer trials, and the cost per quality thresholds that we're seeing come through in recommendations? And is there an issue there that maybe we could marry up with what Deb was saying earlier about bringing forward some of the quality of life data to support potentially patient relevant outcomes? I don't know who wants to tackle that, but we did have some discussions in the prep for this panel around maybe some relationships between the cost per quality thresholds and the surrogate endpoints. What's the link there?
I can go with that. Bob, you seem like you want to.
Pass to you, yes.
I mean, it's almost if you know HG a little bit, it's almost obvious, because almost the vast majority of decisions are based on whether a new drug is deemed to be cost effective, and that we have a simple threshold for that, which is like how many dollars does it cost for an improved for a full year of quality of life improvement. And so if we don't measure quality of life, we will have to guess how the surrogate endpoint impacts quality of life. And best guesstimate is not a great estimate. If we have quality of life data, even if it's sparse, and I think that's coming now with real world data, you can at least extract some of that. There's much more information on that. And so again, it just the quality of life aspect, measuring that, I think personally, I think in a lot of scenarios, it will have much bigger impact than it actually would running clinical trials for 17 years and getting overall survival. Partly because the latter would take so much time, so you wouldn't be able to make a decision for 15 years. So that's just an obvious no-go. But it really is where I mean that, and then I think pharmacovigilance is something that's overlooked as well. Just general safety, which clearly is linked to quality of life as well.
Thanks, Christian. Maybe back to Tara and Lorraine. Just reading your report, notice that some of the the surrogate or non-traditional endpoints were around, the pathology of the disease itself and trying to pick up traces of tumor after a certain period of time. I remember we were working on companion diagnostics as an issue that were coming along in terms of the drug development process. This seems like those companion diagnostics have to carry right through to on market, not just determine which product is going to be used in the first case or at that stage. And any thoughts on the fact that other parts of the whole cancer system are going to have to get ready for this as well in terms of diagnostics?
Yeah, certainly. So while we didn't look at companion diagnostics specifically as part of this research, it is definitely a component of it that's going to need to be taken into consideration, particularly when you're looking at identifying very disease-specific measures for improvement. From an industry perspective, though, Lorraine, maybe I'll hand it over to you to talk about it from that side of things.
Well, yeah, I mean, I think, again, we didn't focus so much on the companion diagnostics piece, but it is an interesting question. I think, you do need to continue to monitor, and that's part of the challenge, even getting to this, the feasibility of even getting to overall survival is because if you're treating in that earlier stage, you're probably going to end up in a place where you're going to have subsequent lines of therapies. You're going to have mandatory crossovers for ethical reasons and the whatnot. So there's so many reasons why that overall survival traditional endpoint is going to be very challenging to meet. So not to mention the, decade plus time to actually get there. So the diagnostics piece will come along. And I think going back to Christian's, remind me of physics class, but all the levers that can impact your disease care, the diagnostics element will come into place. And of course, with that is going to be cost, but then, the flip side of that is hopefully, when you're treating in the earlier stage setting, you're treating with curative intent often, right? So you're hoping that, the investment is worth it at the end of the day, but we do need to be able to make those correlations, of course. I don't know if that answered your question, Bill, but.
It was a question that needs further discussion, but maybe Christian, if you want to jump in there too. I mean, you're doing a lot in the post-marketing setting and RWE, et cetera. And then we've got Louise tagging on the chat. We'll get to that in a second. But Christian, any other thoughts on the diagnostics aspects of this?
Yeah, I mean, there's a lot of different aspects of it. So yeah, I mean, I certainly agree that that's an important one. Can we? I mean, if we just frame it as, is it more the importance of accurate diagnostics, or is it's like the cost of what's the exact question here? Oh,
it's something that we're going to develop and move forward on for future analysis. There's no specific question to you. It was more, I was reading the IQVIA AstraZeneca report, and some of the specific surrogate endpoints seem to require the diagnostics on an ongoing basis, every post 3 months, post 6 months, et cetera. Yes, it would be ongoing cost, but it also seems to marry up well with what Bob was saying in terms of conditional recommendations from CADIF, dependent on ongoing review and research. So, and I know actually everyone on the line is working on issues related to RWE, and that goes back to Louise's question. And what can the broader patient advocacy and cancer community do to support this work, complex analysis, expert working groups, and RWE? I know this panel is going to be taking it on the road to ISPOR and whatnot, and if we accomplish nothing, we're going to just ask some good questions and inspire people to do some really good research. Maybe that's a start. But just with 5 minutes left, why don't we go around the horn what is the takeaway that you want listeners to get from this? Is this an FYI and heads up? Is it a call to action, sort of what I was suggesting in terms of everyone, roll up your sleeves and get at this and bring your expertise to bear? Or is it a bit of both? And maybe Bob, we'll start with you. What do we want to do with this?
Well, a couple of things. So, as Tara and Lorraine pointed out, a lot of these surrogate endpoints are very specific to disease states. So, looking at, I think we have to, if you're representing a particular cancer type, I think it's very, very important that you start looking at this cancer pipeline and identifying that the treatments that are in the pipeline that are going to be, in front of HTA, in the near future, and start to really understand what the issues are going to be with that surrogate endpoint. I mean, we're going to have to wrestle with the fact, is there a decent correlation with overall survival? If not, what is it that you want to do from a, to support for, maybe some sort of conditional reimbursement, and maybe also support, the prospective research in that area, through a patient registry. What we don't want to do is let the whole concept of uncertainty take hold and the drug just to be sidelined without some sort of good plan in place. So, but because so many of these endpoints are very, tumor specific, I think we all have to sort of do our own homework to a certain degree. To Louise's point, though, there's a lot of things we have to do collectively to ensure that CADTH does adapt and develop new methods, and actually takes this very seriously and recognizes that this avalanche is coming and that they have to start moving. And we've started that work already, but I think, we just can't sit back and hope for the best.
Thanks, Bob. Tara, do you want to tag in? Is this a FYI heads up or call to action or both? And, is IQVIA involved in this at the global level too? Is Canada, little Canada starting to lead the way in some of this stuff?
So I'll tackle your first question first, Bill. So yeah, echoing a little bit what Bob said, particularly when we looked at, the right-hand side of Lorraine's, or the right-hand side of Lorraine's slide, where we are looking at the disease-specific outcomes. Those really should be disease and patient-specific outcomes. In there is quite a few, what we would say are more patient-relevant outcomes as part of that, bringing that more to the forefront, as Deb mentioned when she joined us as well, and just being able to improve the acceptance of these endpoints, clearly defining what the endpoints are, validating, having validated instruments to be able to demonstrate that clinical benefit through the correlation between the surrogate and the endpoint, traditional endpoint, as Christian mentioned. Certainly those are all going to be very important. We grouped 55 different endpoints into other endpoints, but those are 55 unique endpoints that are out there in these clinical trials.
Thanks very much. Lorraine, what's the takeaway?
Yeah, I mean, I think a lot of the good points have been made already, but I think just to remind ourselves that this is not a Canadian issue. This is a global issue. All various different HTA bodies are also going to be grappling with this, as well as the regulators. And so, even if we can start to, we need to raise the issue here locally, but even if we can look to other markets, if we can encourage our own HTA bodies here in Canada to work with other markets on how to solve for some of this. A lot of these trials are sit at the global international level. And so we really need to take that sort of global approach. But at the same time, I really want to see Canada lead this. I don't see any reason why we can't. I would love, again, we're here to raise awareness on the topic first and foremost. How do we get to the solutions? I think it's together with all the key stakeholders around the table, but there has to be an appetite to do this. This is complex, as Christian mentioned earlier. There's no easy way through this, but I think we can do it. And the good news is that the science is going there. We're getting to a more curative state, so we just need to keep pushing our processes to catch up with the science, because at the end of the day, it really is about the patients who can benefit from these therapies and from these treatments at that earlier stage setting. So that's my 2 cents.
Thanks, Lorraine. I think that's at least 5 cents. That was great. Christian, last word to you.
Yeah. Last point, just like we said, don't take no for an answer. Don't take uncertainty for an answer. There's too much tendency to say, well, we're uncertain about this, so we're not going to make a decision. We're not going to take any action. There is data somewhere. There is ways we can go and get data. So to Lorraine's point, it's not just Canada that's struggling with this. Let's say there happens to be a small validation study in, I don't know, Belgium. What's to say that we can't use the evidence from there to transport that, as the term is, into Canada and take some, at least reduce the uncertainty. So I'd say anytime someone says, oh, it's too much uncertainty, we can't make a decision, push back, do whatever you can in your position to help them or identify yourself more data, more information that can just help make better decisions on use of these surrogate outcomes.
That's a great way to end it, Christian. And it makes me think of when I first got to know you, it was many years ago, and it was about adaptive clinical trial design. And when I needed to figure out what that was, and then we ended up doing a really good session at CADIF way back in the day. If you need to know about data and how and why trials are sort of constructed as they are, you're a real go-to resource. So thank you, Christian, for helping pull this together and help people make sense of it and catalyze things. And there will be an outcomes report from even the CAPT workshop that we all collectively worked on. So as I said before, the white paper link to that is going to actually be available to all of the registrants for this webinar. We're going to send that to you as a thank you. One final, I'll just pull up this again. Reminder, the Hot Topics Expert Webinar Series is actually online. It's on CanCertainty's YouTube channel and SlideShare pages. So if you just look up CanCertainty YouTube and CanCertainty SlideShare, you're going to find today's session as well as the one on qualities, funding algorithms, and PCPA and provincial listings. Bob, anything else to close it out? Like what's next for this this fun series, if anything, because I think people are really clamoring for more.
Well, sure. I think one thing that's become evident in a lot of my work with patient groups is that there's still, I think, a demand to have a nice, crisper understanding about how clinical trials are designed, also how to actually analyze clinical trial data, and also understanding some of the fundamentals of health technology assessment, things like, what is cost utility analysis, for example. So we're designing some educational sessions right now to sort of like it's kind of back to the basics, but, to give people a good understanding so that when we're starting to engage with topics like this, which are very, very deep and complex, they actually have a solid basis of understanding to work from. So we're designing that right now. So stay tuned. We'll be announcing that in the coming weeks or month or so.
Excellent. And this is CanCertainty. We neglected to show this slide last time, but it's not just one organization, is it, Bob? It's quite a number together.
Yeah, there used to be 35 of us. There's slightly more with a couple of the amalgamations that have happened, with the Canadian Cancer Society merging with Prostate Cancer Canada, et cetera. Our numbers fell by one or two numbers, but we're starting to add a couple new small patient groups as well. But it's a group of about 30 coalition members, along with some affiliated clinician groups as well. So it's a strong, united front to try to achieve equal and fair access to cancer treatments.
Excellent. Well, thank you so much, Bob, for making this possible, as well as the supporters that I mentioned earlier, Amgen, AbbVie, Pfizer, and Merck Canada, AstraZeneca and IQVIA for bringing forward some amazing data here and in other settings. Christian for helping explain very complex causal versus correlative data. It's not easy to bring forward. And Deb, as our guest co-panelist, unexpected. That was really great to have you on briefly. And Bob, thanks for helping pull her up onto the panel. And hope to see all of you again in the coming weeks as things are now open in person. But really thank you to everyone who joined us this afternoon or this morning, depending on where you are. And we'll see you next time. Take care, everybody.
Thanks, everyone.
Bye.